All right, so in this video I'm going to talk about translation, and as a reminder, what we were talking about today is translation, like transcription, includes three steps: initiation, elongation, and termination. Unlike transcription, translation goes from a nucleic acid at the beginning into an amino acid sequence at the end. And also, unlike transcription, translation, the initiation step, does not actually progress past adding in that first piece. Once that first amino acid is added in translation, initiation is over and you transition into elongation. And so, since translation involves a translating from a nucleic acid language or a nucleotide language into an amino acid language, we have to spend a little bit of time talking about how that translation is made, and then also a little bit of time talking about the different players involved. In transcription, we have different proteins, transcription factor 2D, we've got other transcription factors. In translation, we have translation factors, but then we also have several RNAs that are involved in this process. And so this is the genetic code showing you a particular sequence of bases, a particular sequence of RNA nucleotides, and what amino acid those get translated into. And so there are a few things that I want to point out. One is that this genetic code is not universal. The genetic code is actually different in mitochondria than it is in the nucleus, also different in chloroplasts than it is in the nucleus. Many bacteria have a different genetic code, but this is what you would consider to be the most common genetic code. And so a couple other things I want to point out is this right here, this AUG. This is also referred to as the start codon. And so during the process of translation, the machinery will scan your now mature mRNA until it sees this sequence of three bases, AUG. And so not only will that start the process of translation, but then it'll also add a methionine. So every protein starts with methionine, and then that portion may end up being removed later. But every protein primary sequence will begin with methionine until it gets changed for some reason or another. Another thing I want to point out is this arginine, one of our amino acids, is the one that is coded for by the most of the nucleotides, or these three codons, these three base sequences. And then also wanted to point out the three stop codons. So these all basically begin the last step of translation, which is termination. All right, so that's the genetic code. Now we need to talk about the different pieces and the different materials that are involved in the process. One more thing. The third base is oftentimes flexible, and so you can see here for proline, CCU, CCC, CCA, CCG, all code for proline. But then we have some amino acids where that's not the case. CAU here is histidine. CAC is histidine as well. But then you've got CAA coding for a different amino acid altogether. But this third position here is flexible, that sometimes you have that where that third amino acid doesn't bind as tightly, and we'll talk about that. It's called wobble, and it allows for some variability in that third base making up the codon. And so this is the wobble. So here's one of the players involved in this process. This is transfer RNA, or tRNA, and as far as the transcription goes, is very similar to what we saw in transcription of our pre-mRNA. The difference is it has different initiator sequences, different promoters, but and it's written by a different RNA polymerase, but the overall process is very similar to the transcription of pre-mRNA. Now, in this tRNA, you'll notice some three-dimensional shape to the structure as the sequence of your RNA nucleotides actually loop back and form hydrogen bonds with complementary sections, giving us an overall three-dimensional shape of this tRNA, and allows for two really important domains of the tRNA. This one right here, where the anticodon is here. Here are three bases that will be complementary to the codon and will facilitate the translation from that codon, that RNA language, the sequence of three RNA bases, into a particular amino acid. And here's your amino acid binding site on the other end of the tRNA, or here where you actually get an overall three-dimensional shape here. Here's the amino acid binding site. And so this anticodon will go in, and whichever tRNA has an anticodon matching the next codon in the sequence will bind, and when it does, brings its particular amino acid to attach to the growing amino acid chain. And so here's showing you the size of tRNAs, anywhere from 75 to 80 nucleotides. And notice there are 49 different tRNAs, different anticodons. And this was interesting because in the previous slide, you see that there are 64 different codons, because a codon is made up of three bases, and each base there are four options. So 4 to the third power is 64. So there are 64 different codons, and yet there are only 49 different tRNAs. And this is because, again, we have that wobble where that third base is not quite as important. It doesn't bind quite as tightly as the first two positions in the codon to the anticodon, and it actually allows us some flexibility in our different tRNAs. Now, if you remember back to cell biology when we talked about this, or another biology class you've taken, there are actually just 20 amino acids that are used in the human body. So there are 49 tRNAs that bring in or recruit or translate 64 different codons into 20 amino acids. And that's why in the previous chart in the genetic code we saw many of our amino acids being coded for by the same codon. And so here's the size of the nuclear locus that gets translated into this. Notice how its size gets cut down from 497. This isn't the number of bases in the process. This is the number of nuclear loci positions in the nuclear DNA that are involved in making these 49 different tRNAs. Yeah, and so each of these has a unique, that should read anticodon. So each of the 49 tRNAs has a unique anticodon, but may not have a unique amino acid. And so here's this idea of wobble. Here would be normal pairing, where G would pair with C. Your purine would pair with your pyrimidine, forming your three hydrogen bonds. But because of wobble, you get some variation in this, where G, this third position, again, does not bind as tightly. And so you have this G can actually bind or interact with uracil, still another pyrimidine. A purine seems to always have to interact with the pyrimidine, but not the pyrimidine it typically interacts with. And so what this allows is for some flexibility in what amino acid you can get because of which tRNA can actually bind to the codon. And so in order to actually have a mature tRNA, it has to be charged. And so, the tRNAs, when they're transcribed, they do not leave the nucleus containing the amino acids. Instead, what they do is each tRNA, or rather each amino acid, has a specific tRNA synthetase enzyme that it binds to, and this tRNA synthetase attaches that amino acid, in this case is valine, to any of the tRNAs that can actually bring that amino acid in this process of translation. So every time a tRNA is involved in the process of translation, remember it donates or brings or transfers its amino acid to the growing polypeptide chain. And so as this happens, it now lacks its amino acid and must be recharged in order to be involved in translation again. And so this enzyme, or several enzymes, one for each of the amino acids, works to charge the tRNA. And so here's an image of a charged tRNA, a tRNA that has its particular amino acid attached to it. Here's your amino acid here. Here's this R group, which is a variable portion of the amino acid. And you can see here's our tRNA. Here's a ribonucleotide with adenine attached backwards to this complex, and then that's where the tRNA gets charged, adding the amino acid. And then the last role player, if you will, the last machine that we're going to talk about before we actually talk about the process of translation is the ribosome. And so ribosomes are what are called riboproteins, and this is a complex of proteins along with ribosomal RNA. And what's interesting about ribosomal RNA, sorry, let me go back here. What's interesting about ribosomal RNA is it is catalytic, and so, or enzymatic. You have a number of proteins that functions as enzymes, catalyzing reactions, lowering the reaction rate. And so once people knew that ribosomes were made up of both proteins and nucleic acids, most people assumed that it was the protein that was actually providing the catalytic activity of transferring the amino acids, breaking that bond between the amino acid and the tRNA, and facilitating the formation of the peptide bond, that it was one of the proteins that was involved in that. But we since have discovered that it's actually the RNA that is catalytic. And so, yeah, I mean, it's unique and is something that's actually fueled an idea called the RNA world, which we'll talk about on Wednesday. And so here's the process of initiation. Here you can see that during the initiation, both ribosomal subunits actually come together. And so here we have our small ribosomal subunit, the 30S unit. During, again, still during initiation, you get your initiation factors 1, 2, and 3. You get a GTP, which is guanine triphosphate. You get your mRNA sequence binding to your small ribosomal subunit at the Shine-Delgarno sequence, a string of nucleotides showing the small unit where it can bind. And then here, once your methionine, your initiator tRNA, that has the anticodon for AUG, which is the start codon, once that tRNA binds to the process at that start codon, it will facilitate the transfer or the attachment of the large ribosomal subunit onto the small GTP that will be broken down into GDP to provide the energy necessary to bind the large ribosomal subunit on. Transcription factors 1 and 2 will fall off. Transcription factor 3 fell off after this methionine binded to the anticodon, and now initiation is complete. And again, I said unlike initiation of transcription that continues all the way until you've transcribed past the promoter, past that initiator. Initiation of translation is complete once this first translation is made, once you translate from that start codon into your first amino acid. And then, so here just labeling some of the different components. You have your A site here, which stands for amino acid site or amino site. This is where your new tRNA is going to come in. Here's your P site or your polypeptide site or your peptidyl site. And this is where you have your tRNA that is physically attached to the entire polypeptide chain. During initiation, we only have one tRNA here, and it's in the P site because this is our entire polypeptide chain made up of a single amino acid. And then here is the E site, ejection site, or exit site, and the ribosome will pull this mRNA through the ribosome, shifting in this direction here, shifting the position of these tRNAs from the A site initially over to the P site, and then to the E site where it is ejected. And so here's elongation, and elongation will continue as long as a tRNA can bind to the next codon by matching up its anticodon. Remember, in this third position, we have some wobble, some flexibility. So the same tRNA can actually bind to possibly more than one codon. And so, again, the RNA, this is peptidyltransferase, the center here. This is a catalytic ribosomal RNA. And so this physically moves the amino acid onto this new amino acid, forming a peptide bond. And then at that same time, your ribosome shifts down, or it actually physically pulls the mRNA further, and you get the opening up this A site, and a new tRNA can come. And so here now your initial tRNA, the methionine tRNA, is now in the E site, the ejection site. It's ejected. Those hydrogen bonds are broken. The tRNA is released, and then at the same time, a new tRNA enters into the A site. And so this will continue all the way up until you get to one of those stop codons. And the stop codons don't have a tRNA that binds to them. There's no tRNA with an anticodon to match a stop codon. Instead, there are 3 release factors, which are proteins that have the ability to bind, to recognize, and to bind those stop codons. So again, there's no tRNA to bind there, and so instead there are proteins that recognize and bind those termination sequences or the stop codons. And when that happens, there's no amino acid sitting in this position for this polypeptide chain to be transferred to by peptidyltransferase, and so the whole complex becomes unstable and falls apart. So here are your 3 stop codons again: UAA, UAG, and UGA. And so all of those, again, do not have a tRNA with an anticodon for them. And so when one of these release factors binds there, the process can't continue. Peptidyltransferase can't do its work. Therefore, you can't move the ribosome forward. Therefore, the whole process falls apart. And now the small and the large subunits can be used to translate a different mRNA, or even the same mRNA, but back at the initiation site. All right. And so in reality, you don't have a single ribosome operating on any one mRNA. Instead, you have several ribosomes operating on the mRNA at any one point. with various length polypeptides. And see, this one is not far away from that start codon, and so the polypeptide sequence is very small. And then here you're getting towards the stop codon, and so your polypeptide is getting close to its maximum length. And then this ribosome here has already reached a stop codon. A release factor recognized that, bound to it, interrupted peptidyltransferase, and led to the deterioration or the disassembly of the entire complex. And now these subunits again could be used to come back here and just continue this process over and over and over again. And so you can see from a single mRNA, you can generate an enormous amount of protein product. And this is the reason why all of this needs to be highly controlled by the cell, because gene expression and the amount of it is an incredibly important process to regulate. So yeah, each mRNA will be translated many times before it's degraded by a nuclease in the enzyme. All right, very few proteins are actually translated completely inside of the cytosol. Many of these proteins have to operate in particular places and aren't labeled for operating in the mitochondrion, or aren't labeled for operating in the nucleus, and instead are meant to operate within the rough endoplasmic reticulum, or within the smooth endoplasmic reticulum, or within the Golgi body, and so, or on the cell membrane itself. And so any of those proteins that operate there actually need to end up inside of the rough endoplasmic reticulum for processing and transport. And so here you can see a mRNA that's being translated by multiple ribosomes, and these ribosomes are physically attached to the rough endoplasmic reticulum. So as they translate this protein and generate this this polypeptide, it is actually shuttled into the endoplasmic reticulum through the membrane as it's being translated, and then once it's inside here, it can be processed. It can be packaged, and it can be shipped to where it needs to go. All right, so that basically is the end of translation, but there are some post-translational modifications. In fact, it's very common for your primary sequence, your polypeptide, to be modified in many ways before it becomes a functional protein. And so here we have our growing polypeptide during the process of translation. Here we have the polypeptide separated after a release factor binded to the stop codon. And then here we have three different post-translational processing or post-translational modifications. Here you have proteolysis, where this polypeptide is cut down. Some of these may be completely recycled, but some of these may just be smaller polypeptides. Here you have glycosylation, where you have sugars added onto particular amino acid residues in this polypeptide. Here you have phosphorylation, where you have phosphate groups added onto particular amino acids in this polypeptide. And another post-translational modification is just helping the protein to fold correctly. And so some additional methylations, sometimes you'll add a methyl group to amino acid residues in the polypeptide, or myristoylation, which is adding a section on the protein to allow it to hydrolyze myristoic acid. Ubiquitination, adding a ubiquinone onto the protein to modify it. Sumoylation, adding a ubiquinone. This is small ubiquinone-like modifier, and adding an entire complex add to the polypeptide to change and help to control its function. And so there are a number of different ways in which you can modify these proteins. And so when you reflect back on this entire process, you'll remember that in transcription, we generate a pre-mRNA that can be spliced in several different ways, generating a number of different mature mRNAs. And then these mRNAs can be modified after they're translated into protein, can be modified in several different ways to generate, again, a large variety of different proteins from the same sequence within the genome.